Mendelian genetics. Introduction. What are Mendelian genetics? Mendelian genetic is an inheritance pattern that follows the laws of segregation and independent assortment, in which a gene inherited from either parent segregates into gametes at an equal frequency. And who discovered it? Gregor Mendel. He was born on July 20, 1822, in Hinces, Vresen, Czechia. He was an Austrian biologist, meteorologist, mathematician, Augustinian friar, and abbot of St. Thomas's Abbey in Birkenau, Brunn, Margraviate of Moravia. The Mendel's laws. He studied inheritance using pea plants from 1856 to 1863. He also conducted experiments with over 28,000 samples and pioneered the use of statistical analysis in biology. There are three Mendel's laws, which are the law of dominance, the law of segregation, and the law of independent assortment. The law of segregation states that the two alleles for each gene are placed in different gametes. The law of independent assortment states that the inheritance of one gene doesn't affect the inheritance of any other gene. The law of dominance states that when two different alleles are present, only one is dominant and will be expressed. Introduction terminologies. Alleles. Alleles are one of a number of alternative forms of the same gene occupying a given position on a chromosome. Heterozygous. Heterozygous of an organism that has two different alleles of a given gene. Homozygous. Homozygous of an organism in which both copies of a given gene have the same allele. Law of dominance. The genotype of homozygous dominant represent as both capital A, for both lowercase it represent for homozygous recessive, and combination of uppercase and lower is heterozygous genotype. What is law of dominance? Mendel's law of dominance states that in a heterozygote, one trait will conceal the presence of another trait for the same characteristic. Rather than both alleles contributing to a phenotype, the dominant allele will be expressed exclusively. It uses monohybrid cross, which is the single trait. Only one form of trait appear in the next generation, dominant trait in the phenotype. Traits are controlled by two factors, which are the dominant, or recessive, expression of only one of the parental character in F1 generation, and expression of both in F2 generation. Law of segregation. The law of segregation uses monohybrid cross, the single trait. It uses monohybrid cross, which is the single trait. Each allele segregate into separate gametes, stabilized by metaphase 1. Each individual has a pair of factors controlling each trait, and one inherited from each biological parent. During the formation of gamete, these two factors separate. Only one allele ends up in each sex cell. In homozygous parents, all gametes produced are similar, while in heterozygous parents, two kinds of gametes are produced in equal proportion. For P generation, each homozygous parents in the parental generation forms only one kind of gametes. Yellow parent forms yellow gametes, and green parent forms green gametes. In F1 generation, the heterozygous F1 offspring forms two kinds of gamete, which is combination of both parents. F2 generation is self-pollination of the F1 offspring, and forming F2 offspring with a 3:1 phenotypic ratio of yellow to green seeds. When the gametes join to produce F1 generation, all offspring of homozygous dominant and homozygous recessive parents are heterozygous. The heterozygous F1 individuals can put either a dominant or a recessive allele in each of their gametes. A Punnett square is one way to predict the outcome of a cross by showing all the possible combination of all the gametes. Law of independent assortment. This law states that genes for different traits are inherited independently of one another. During the formation of gamete, sperm fuses with egg. The alleles for each gene segregate randomly and independently. So, the inheritance of one trait does not affect the inheritance of another. This principle lead to genetic variation in offspring. Dihybrid cross. It is important to study the inheritance of two traits simultaneously. An example of P-plant model is used to demonstrate this dihybrid cross. First, homozygous dominant with smooth and yellow seed and homozygous recessive with wrinkled and green seed is crossed. Then it will produce F1 generation, which has heterogeneous characteristics. Then F1 generations self-pollinate, where the gamete combine randomly to produce an F2 generation with 4 phenotype ratio of 9:3:3, one which are 9 individuals with round, yellow seeds, 3 with round, green seeds, 3 with wrinkled, yellow seeds, and 1 with wrinkled, green seeds. As we come to the end of this discussion on Mendel's work and the fundamental principles of genetics, it's important to reflect on his incredible contributions and their impact on the field of biology. Mendel's meticulous experiments with pea plants led to the establishment of 3 foundational laws of heredity: the law of dominance, the law of segregation, and the law of independent assortment. These laws provided the first scientific explanation for how traits are passed from one generation to the next. His use of statistical analysis in biology was groundbreaking and set a new standard for scientific experimentation. Although his work was not fully appreciated during his lifetime, it became the cornerstone of modern genetics after being rediscovered in the early 20th century. From Mendel's experiments, we learn that genes exist in alternative forms called alleles, which influence the traits we inherit. The predictable inheritance patterns he described still hold true for many traits, even though we now understand that some traits involve more complex genetic mechanisms. Looking forward, Mendel's principles continue to guide genetic research. However, modern advances like molecular genetics, epigenetics, and polygenic inheritance are adding layers of complexity to our understanding. These developments remind us that while Mendel's work was revolutionary, science is always evolving. In summary, Mendel's legacy is not just his laws of heredity, but his approach to scientific inquiry, using careful observation, experimentation, and analysis to uncover the principles of nature. These are the references used in this assignment. Thanks for watching.